Hello. So we'll be working on the practical applications of the introductions you know learned at the curriculum one. So we'll be moving to curriculum two, which is multiple sequence alignments, and we'll be dealing with the practical base. So this is multiple sequence alignment, and in multiple sequence alignment, which is an extension of pairwise alignment to incorporate more than two sequences at a time. So this time around, we are actually dealing with multiple sequence alignment, involving alignment of more than two sequences. So pairwise alignment is just two sequences, but this time around we're dealing with more than two sequences, and multiple alignment methods try to align all of the sequence in a given query. So multiple alignment are often used in identifying conserved regions across a group of sequence hypothesized to be evolutionary related. So we have a lot of sequences, a group of sequences, but then we are looking for to identify conserved regions. We are talking about conserved regions. We're talking about regions that are highly similar. You understand what I'm saying now? That are highly similar, that have compatibility. When it comes to resemblance and similarity. Now we known that such conserved sequence motif can be used in conjunction with structural and mechanical information to locate the catalytic active sites of enzymes. So it is possible that once you align a specific enzymes, you have similarity, resemblance when it comes to the enzymes that are contained in a sequence and other sequences. So we use multiple sequence alignments to understand the certain enzymes that exist in a sequence and other sequences at the same time. So we have alignments which are also used to aid establishment of evolutionary relationships by constructing phylogenetic tree. So we're talking about understanding the same ancestors. You understand what I'm saying now? Like different organisms coming from the same ancestors, sharing the same ancestors. Of maybe you want to talk about sequences that are highly similar, or maybe you have a lot of sequences and you want to check which one are highly related, when it comes to evolutionary relationship, that maybe they share the same ancestors, the same percentage of similarities, and then and a lot of things. So let's just look at what phylogenetics is all about right now. Don't worry, we'll look into the practical application of this section, but just want to give a brief introduction of what we're actually working on. Now we have phylogenetic analysis. Now phylogenetics and sequence alignment are closely related fields due to the sheer necessity of evaluating sequence relatedness. So phylogenetics and sequence alignment are closely related. So you can't do phylogenetics except you actually work on what's it called now, on alignment. So before you can begin to build tree, understand phylogenetic relationships, difference and divergence in species, in genes, you understand what I'm saying now. So you just have to first run sequence alignment, which is multiple sequence alignment actually, so that you'll be able to work on the phylogenetics and get the necessary result you're working on in the research. So then the field of phylogenetics makes extensive use of sequence alignment in the construction and interpretation of phylogenetic trees. Yeah, the field of phylogenetics makes extensive use of sequence alignment, to make a lot of constructions, phylogenetic trees constructions, you understand, and used to classify evolutionary relationship between homologous genes represented in the genomes of divergent species. So we have homologous genes, you understand, that exist between different sequences, and in different diverse sequences, that are related to different organisms. But these sequences actually share a common gene. Maybe they have like 70% similarities, 80% similarities, 90% similarities, maybe even between sequences from different organisms like man, different animals like rats, like maybe rabbits and things like that. So in a way that these species are divergent, but then tends to have common, relatedness when it comes to homologous genes. Now, the degree to which sequence in a query set differ is qualitatively related to the sequence evolutionary distance from one another. So, roughly speaking, high sequence identity suggests that the sequences in question have a comparatively young, most recent common ancestor, while low identity suggests that the divergent is more efficient. So, once run if the phylogenetic trees of multiple sequence which you have aligned, so those who are identified share high sequence identity proceeds to actually have recent common ancestors. You understand what I'm saying now? Recent common ancestors. Why those who show a low identity when it comes to comparison, when it comes to resemblance. You understand? So that means they have their divergence. You understand? And more ancient. That means their ancestors are more ancient. Because over time they might have developed certain traits. Certain characteristics, certain traits, certain behavior. You understand? You would deem them that is kind of different from the ancient ancestors. So that's some of the essence of phylogenetics. Now let's see one other thing about phylogenetics. Now we have knowing the evolutionary relationship among species allows scientists to choose appropriate organisms for the study of disease such as HIV. So maybe you're actually working on HIV and maybe working on the live experiments of clinical trials of HIV. You understand? So all you need to do is check and look into certain organisms. Or maybe maybe you want to work or try it on mouse. You understand? So or different organisms. So you can actually use evolutionary relationships when it comes to building tree to understand those organisms that are closely related to man. You understand that share common ancestors. You understand what I'm saying? So that you'll be able to work on it, because there is probability that you work on an organism that is not in a way related to man. So as such, even once you run the clinical trial or you do a lot of things, so it might yield results in a way, but then there must be highly differential rate and range between those two organisms, because by the time you finish dealing with that organism and you proceed to man, there will be a very wide range of differences that can result to different issues, a lot of issues. So it is highly recommended that you work on evolutionary tree to be able to know sequence or maybe organisms that are highly related to Homo sapiens, so that you'll be able to have good results, and and then your results may be narrowed to maybe 80% of result achievement within those two sequences. That means mouse and man. So scientists are even using the principles of natural selection to identify new drugs for detecting and treating diseases such as cancer. You understand? So they use principle of natural selection, because in phylogenetics we have natural selections, we have different dimensions of evolutions. You understand now? So natural selection is actually part of it. So they use natural selections when it comes maybe plants, relating to plants, and I just use it to identify new drugs for detecting and treating disease such as cancer and so on like that. Now we have scientists separate strands of DNA from both species using it, which is denaturation, which breaks the bonds between the base pairs that link the two sides of the double helix. Now parts where base pairs links based upon exhibit genetic similarity, the more information that links up, the closer the species evolutionary. So that means once you get data from NCBI databases, and then you get data of different organisms, and then you align them together, and then building the tree, the genetics analysis. So the sequences that show genetic similarity actually shows that those species are closely related evolutionarily. So that's one of the essence of that. So Now, can we just look at the tables? We have 5 stages of molecular phylogenetics analysis, which is sequence acquisition, multiple sequence alignment, substitution methods, tree building, tree evaluations. So I'm going to be working on this right now. So just follow me. Now, presently, now we want to work on multiple sequence alignment. So after we're through with multiple sequence alignment, you can proceed to phylogenetics analysis. So that is the essence of this. So we're gonna run this, so just follow me very carefully as you see this now. We actually been working on a particular data, which is the hemoglobin beta gene of that results to sickle cell problem. That is the gene that exists, that took place in hemoglobin in the body, in the blood. So all we need to do right now is we just need to look for variations, you understand, of this sickle cell, this mutant cell on the database. But these sequences actually belongs to different patients, so there's probability we have a lot of variations in hemoglobin beta genes. Variations of hemoglobin beta genes from different patients. That's what I'm saying now, from different patients, because there's probability that the mutation that took place that result to sickle cell in a particular patients can a little bit be different in diversions. You understand what I'm saying now, with another patient. So we want to look at variations in this mutant gene, different variations. So that's one of the things we want to look into. So yeah, just follow me very carefully as we explore this. Now, the first thing you need to do is you download the different hemoglobin beta gene data from the database. But what you need to do is you blast, because you already have your query, the one you are working on, the original one. So there are times, maybe for instance now this coronavirus, so we can actually look into the variations of coronavirus in Nigeria, compare with every other coronavirus sequences from different states, from different countries now, even compare with the one from Wuhan, which is China. So what we need to do is we sequence the coronavirus in Nigeria, and then we use that sequence to run a kind of maybe multiple sequence alignment, and then we look for variations, you understand, look for, you can even build tree to understand the particular coronavirus that is highly related with the one in Nigeria, because at some point, as those as the coronavirus begin to infect one patient or the other, variations arises, you understand, mutations arises, and used to different issues, and then features of coronavirus maybe at times can even mutate to advanced coronavirus. So we want to work on sickle cell right now, so you can actually work on coronavirus if you want, based on our research actually. So let's go to the applications now. Mind you, we have a lot of applications here, which is this and this. So we're still gonna work on it, but let's work on the sickle cell, which is this. But then this particular exercise, which is one, is for pairs alignment, but we want to work on multiple sequence alignments of this. So just follow me very carefully, very carefully now. So we're working on applications. So what you need to do is go to the NCBI database page. So you go to blast, which is this place, which is here. So you click blast. Right. So what we're working on, we are working on nucleotide sequence. So we work on nucleotide blast, which is nucleotide blast. So you click it, because we need a lot of sequences that are actually in hemoglobin beta genes, because we want to look at variations, we want to study the phylogenetics role when it comes to that. So now what we need to do is you choose the file, because it's already on your system. The hemoglobin mutant gene now, yeah. So here's mine. So in case you don't know what the folder, just look for the folder. Mine, and name it sickle cell. So human normal gene and human HBB mutant gene. So we're actually working on HBB mutant gene. HBB mutant gene. We want to look at variations. We want to understand differences. We want to work on multiple sequence alignment of this mutant gene, and look for the work on phylogenetics analysis. So in terms of looking for variations, you understand what I'm saying now? Differences, similarities, and differences when it comes to both. So you just open, so it's already there, which is this homologous VB gene. So you keep going. So we're working on a standard databases, which is nucleotide collections. Yeah, so you just keep going, leave the rest that way. So you look at this. So maximum target sequences. So let's actually pick let's pick 50. Let's pick 50 and see. So you blast. Yeah, so keep going. 2 seconds. Yeah. So here's it. As you can see, we have like 50 results, a whole lot of 50 results here. So as you can see here, selected 50 results. Now actually these are query sequence, this is the original sequence because it is 100% similar. Now we begin to have 99.94, 99. All these are hemoglobin HBB mutant gene. Yeah, so all this, but they differ, you understand, in variations, you understand, in differences they are different, as you can see, very, very different, as you can see. So what we need to do is let's like download maybe, because 50 will be should be too much, so let's work with 15. Let's work with 15 sequences, including the original one, the actual one, and these are query sequence. So whatever you will be working on when it comes to phylogenetics after the multiple sequence alignment, just make sure you understand that this is your query sequence, this is your original sequence. So you just keep going. Now we have 99.94. we have a lot of 99.94, a lot of them. We have a lot. But just 3. Okay, so you can just keep picking, pick. Keep picking. So you pick like 15. That's 12. So until it is like 15. So I think we have 15 now. So including a query sequence, which is this, that's 15. So you download, you just click here, faster complete sequence. So as you can see, it's downloading. I think it has downloaded, so you can check your downloads. Yeah, so here's it, and you open the folder, showing folder, you click. Yeah, so here's it, and you rename it to multiple mutant gene. So and you save. So as soon as you're done, you cancel and you go to your Geneious software. Just goes directly to your Geneious software, and then you create a new folder, which is multiple multiple sequence alignment. So multiple sequence alignment, and you click, so, and you create another folder, phylogenetics analysis. So that's about 2 folder, but we're working on multiple sequence alignment first. Then you import the folder you downloaded from NCBI, the multiple sequence you download. So it's on our desktop, and it's named multiple sequence. Here is it. So you import, then you keep sequence separately. Keep sequence separately. Yeah, so you have all what you actually downloaded. You have all what you actually downloaded here. You have all what you actually downloaded. Now mind you, you must take notice of the query sequence. What's the name of this query sequence? So here is the session number. You know the name of the query sequence by their session number, which is AU7615. Then you go back to the to the Geneious software, AU7615, and you look for it. AU7615, here's it, and you can now rename it, edit name to query sequence, query sec. You understand now? Yeah. So now remember, we are working on multiple sequence alignment. So a multiple sequence alignment is aligning sequences that are more than two. Are you getting it now? So all you need to do is you pick everything here, all the sequence you downloaded here, because we want to run the multiple sequence alignment of these sequences. Now you just click here and double click, so everything will be aligned. You understand? It will be picked. Everything will be picked. So as you can see, everything is picked here. Everything is picked. So just go to align and assembly. As you can see, align and assemble. So you just click, then you run your multiple alignment. Yeah, that's it. So you use Geneious alignment. Now we are a lot of tools for sequence alignment on Geneious software. So we have Geneious alignment itself, we have muzzle alignment. So these are cluster alignment, these are alignment tools. You understand now? Alignment tools, translation align, and mapped alignment, cluster omega. All these are sequenced alignment. You understand now? But you can actually choose anyone, but then I myself prefer Geneious alignment because it's provide you with simple step. Just like as if you click here, you can see these different options, but then you can use Geneious alignment. Now in Geneious alignment we have global alignment with free grabs. Remember, we have different types of alignments. Let me quickly show you that. So we have types of alignment. So we have pairwise alignment, multiple sequence alignment. So we're actually working on multiple sequence alignment, but we have alignment methods as methods of alignment. And in methods of alignment we have global alignment and we have local alignment. And in global alignment, which attempts to align every residue in every sequence, like that is every sequences in all the sequences we just downloaded now, it aligns them one after another. You understand? One after the other, which attempt to align every residue, you understand, in every sequence, as most useful when the sequence in the query set are similar and roughly equal size. Now we have local alignment, are mostly used for dissimilar sequence that are suspected to contain region of similarity to similar sequence motifs within that larger sequence motif. So these are actually, the local element is actually used maybe for sequences that are very far from themselves when it comes down, or maybe that are divergent. You understand? Is actually wide. You understand what I'm saying now? So that's it. So we'll be working on global alignment, since we're working on multiple sequence alignment. So global alignment with free gaps and hens. Now the essence of with free gaps and hens is talking about once you're aligning the sequences, like the beginning and the end of the sequence, you have a free gap. You understand? Like it should kind of be free. Like you're just trying to say alignment should not occur in those free gaps. So you're actually looking for a conserved regions that are highly similar for alignment and comparison. Now cost matrix is talking about the percentage of similarity you're actually looking for. So but this time around we are looking for identical. If I'm talking about identical, I'm talking about 100%. So it will give you a wide range of similarity check when it comes to 100% checkup. You understand that? 100% measure. So I think we're through with these more options. You can just gap open penalty, leave it that way. Gap extension penalty, leave it that way. Refine iterations, just leave it that. Now gap open penalty is just trying to make you understand, or you're just trying to tell Geneious to align that the gap the percentage of the gap when it comes to the alignment procedure should not be more than 2 when it comes to dissimilar sequences. Now the gap extension penalty, like the extension penalty of this gap open penalty, that it should, okay, if at all it wants to extend, it should not be more than 3. So that's just the essence of that. So then you run, so it's going Geneious alignment, okay, still going, wow, 0.014. so I think it's going to take a little bit more time. So give me some times, we're going to be back in a short while. Yeah, so the alignment has finished running. So here's the alignment. So you'll find something like this, nucleotide alignment on your system, so which is alignment of 15 sequences. So this is the view of the alignment, as you can see here. So we have all the graph, all the displays, just like what we have done the other time. So, and then we have the consensus, which I believe you understand what consensus is. This is the consensus. Now the consensus determines the rate at which it determines the similarity, or the measure of the similarities of the sequences that each nucleotide sequence are supposed to have. You get what I'm saying now? So that is the essence of consensus identity. So the actual nucleotide bases are supposed to be equal, they're supposed to be here on in the nucleotide sequences, is actually what we have on this consensus. And consensus also determines the rate of alignment, and when it comes to similarity and differences between sequences. So once you have differences in elements, you have a gap like this, but once you have similarities and elements, you have something green like this. As you can see, green is 100%, while green brown is at least 30% on your screen, under 100% identity, and then red is below 30% identity. As you can see here, we have a lot of gap, there are a lot of reds here. So just follow me as I try to give you. So you just try to zoom, and then try to adjust it. Now as you can see, we have a lot of long sequences. Now the essence of this, the reason why all this is very long is because of this actual sequence. Now remember, we have our query sequence here, and our query sequence is number 2, while this actual sequence here. Now as you can see here, this sequence here is like 207,346, as you can see here. So that is why you say it's very long this way, but every other sequences, they have like very short, which is about 1,557 here, 1,000. So just between the rate of 1,000 something, 1,000 plus, and then we have 3,000 here or 2,000 here. So this sequence actually long. So that is why you see we have a very long extent of alignment here. But the first alignment, as you can see, is actually not working here. So our query sequence is actually this, which is this red, this black alignment here. So, and then we have this long one, as you can see, very long, but our region of interest is just here. It's just here, as you can see. Now let me quickly show you something. In alignment, there's something I really want to show you here. So maybe let me just click multiple alignment. Now you see, with free grab and end, so had it been I choose global alignment, which is without free grabs and end, so actually it will trim all those side, this side. Let me show you. It will trim this long whatever that is seen here, to trim the whole of this place to this place, and then trim the whole of this like this. It should trim it down to the last place, because we are not interested in those areas, so we don't really need it, as you can see. But this is actually our region of interest, as you can see. Just this place, this is where I liked it. So actually it's our region of interest. So all you need to do is just zoom, so we understand what we are really working on. So as you can see, we have some little greens line, like I'm talking about the consensus now. So you just kind of zoom very, so that you have, as you can see here, this is actually showing a huge range of differences that is amongst all the nucleotide sequence. Now each of these nucleotide sequence has a line, as you can see, a line here, a line here up to this, last two. So all these have a lines, but this just trying to make us understand which is the consensus sequence, that a whole of this region from here to here actually have a difference. Like every of the sequences, all of them have huge differences from here to this place, but from here to here all of them have the same sequence. So for you to just see, you can just zoom, so that means we have CTs. That means the whole of these regions carry CT, which is entirely all the sequences, so they all have CT here. So we have G, so that means the whole of this region here, there is a lot of differences. So that means only these two sequences actually carry the same, whatever I can share now, the same nucleotide sequences. So every other one from here down to this, actually they are different. So that's why you see these differences. So anywhere you see this, this red bar actually shows consensus, which means from here to here all the sequences actually have the same nucleotide basis. So that's the meaning of that. Now there is something I also would like you to understand. So but first, as you can see, all these places, all of them, see, there are a lot of differences here. Now there's something I want you to understand. That's what we call conserved, semi-conserved, and a lot of things. So all these regions where we see this red bar, which is consensus, shows that all these places are conserved. These places are really not conserved. This place is conserved. This, and we call this not conserved, as you can see now. So that's one thing about multiple sequence element. You can make more research to have deeper understanding about that. So actually, as you can see, this area to show the whole of those sequences. So this area we've been talking about actually don't have any similar sequences with others. Only these places, which is where let me just try to. So as you can see here, this place actually conserved because we have CT all over, just like what I was saying. So they are only conserved, but here it's not conserved. Is not conserved at all in a way. So but this is actually conserved. So this also conserved, as you can see, it's conserved. Yeah. Now the reason why it's actually really not conserved with everything here, the reason all these places are not conserved because of this last one. And maybe this last one also have the GCCAAT. So that means it's actually conserved. So that's the essence of that. And so on. So all these places, had been you pick the other global element without free gap, which I just showed you. I mean this now. So you won't really have all these lines. So we're talking about now. The next thing you are to move on is to build your tree. Yeah, guys. And if you are building phylogenetic tree, that means you are running your phylogenetic analysis. So don't forget, I told you cannot run phylogenetic analysis until you have done multiple sequence analysis. So since we have already done our multiple sequence analysis and we are on it, so we can actually build our tree. So the building of the tree is actually the phylogenetics analysis. So you just click, as you can see, you click it, and then so we have Geneious tree builder. We have a lot of tree builder, just like alignment tools. So we have tree tools here too. So just like you can see, Geneious tree builder. So just use the Geneious tree builder and leave it this way. Neighbor joining, you leave it that way. Then no out group. Now choose out group. You just have to choose out group. So as you can see, we have the number one is so very long. Now this out group, actually it is that you need to include a sequence that is very far away in similarities from all these sequences, very far. So that's why we're picking this number one, not this. We're actually picking number one to be our out group because it's actually very different from this. So whenever you're working on tree phylogenetics analysis, you need to really pick an outgroup, like a sequence that is very very far in similarities with the sequence you're actually working on. Maybe just if you're working on sequences on animals, you can pick a sequence of plants so that it can really be far away. The essence of that is just for you for the Geneious software or the analysis to be able to come out accurate. So you just pick this, because since we actually have it to be very very long, which is the actually the first sequence here. So that's it, and then you click OK. So the next thing, you click resample tree. So this is very very important now, so just listen now. This bootstrap, you click it. We are using bootstrap. Now the essence of this bootstrap is just to give identical probability number, statistic numbers for us to be able to run the phylogenetics very according to what I'm going to be telling you now. The random seed now should be 1000. Whenever you're conducting a phylogenetic tree, always put your random tree to 1000, as you can see, while the number of replicas should be 100. Now create consensus tree or sort of topology is just an option for you to be able to run your tree in order to pick accurate results, to have accurate results. So just you can use any of these and leave it to maybe you hear 50 or so, but try always to be using create consensus tree, because if we're talking about consensus, we're kind of talking about similarity. So but what we're working on is not the similarity. No, we're working on similarity. So you click OK, and it's gonna run. So as you can see, these are tree. So that's build our tree. So this is the out group we initiated here. This is the out group, and all these are now these are query sequence. These are query sequence. As you can see, this is our query sequence, and then that means this is trying to tell us that the whole of these are 100% identical. Now remember that we have what we call gap penalty, which is 15, which is 10, and then we increase it to 3. Which is 12. We increase it to 3, which is 15, and then so this is trying to tell us that it created 15% gap penalty, so that at least any similarities beyond that 15%, it is actually not 100%, but in as much as it's within the range of 15%, so that means all these sequences are 100% similar. So with our query sequence, but this is out groups, which actually very very far away. But at the bin, we have a different kinds of sequences. Maybe we have like 60, we have 70%, we have like 80, we have like 90, just like that, in variables. So that means this telling us that this particular graph will be telling you maybe this place kind of been like maybe 60 here, 70, and things like that. So once you use this to run an iridia, which is your assignment actually, so you will understand what I'm trying to say. So that's that, and if you have questions you can ask. So now the essence of this is that all these are similar, and they all have, because this is actually the root, so that means they all have the same ancestor. They all have the same ancestor, as you can see now. So they all have the same ancestor, and they all have the same evolutionary difference. Similarity, not difference. Now they all have the same evolutionary similarities. So that's the essence of this now. And you remember that we're working on sickle cell, so that means all these sickle cell, the variations are actually not much. So that means they are highly similar, though they are all from different patients, but then it's as if the rate at which the mutation occur is actually in the same maybe region. You understand? Mutational region that that exists between all the sequences. So that's the essence of that. So you can actually choose to do some things which is so time labors, do some editing. So you can actually go through this. Now we have different formats of tree. You can choose this, so you can expand, try to expand, try to really expand. So as you keep on, then you keep having something like this, as you can see, keep having something like this. So you can even use this, so which is actually showing us this. So it depends on whatever you want to use. whatever form of phylogenetic tree you want to use. So it depends on you. So we have come to the end of multiple sequence alignment and phylogenetics analysis. Please make a personal research, on what multiple sequence alignment is and phylogenetics. Make series of personal research, please. So thank you very much. Now, please, before we round up, I just want to give you another clue. So had they been all these are maybe different, maybe an organism which is an animal, but in the same maybe ancestor, maybe different species of rats. So and then you want to work on drug, you're working on drug, maybe clinical trial, and you get all these mouse and you're trying to understand the phylogenetic relationship within them, and maybe work on natural selections. So this will give you understanding that you can actually use any of these, maybe mouse, if had it been all these sequences are mouse, so tell you that you can actually use any of these mouse to run, because they all have 100% similarities. You understand what I'm saying now? So that's it. Thank you very, very much.